Home stretch. You can do this. Let's go. All right. So, yeah, there's a bunch of proteins we're going to learn about. Let's go learn about them, and then we'll see which ones are super duper important. So now we're talking about B cell receptors. And so you remember the parts of the B cell receptor? You're going to have your thing that looks like an antibody, which is the B cell receptor, made of 2 heavy chains with an extra little transmembrane region on the bottom, and 2 light chains, as in a regular antibody. Do you remember what those 2 little things next to them are called, with the little ITAMs on the bottom? Yeah, those are going to be Ig alpha and Ig beta. All right. So once we've got our complex there, we are able to bring over, so we're able to make one of these Sarc family kinases, and there are several of them, and so you can just remember them as a Sarc family kinase, SRC. And the Sarc family kinases are going to phosphorylate the little tyrosines on our ITAM motifs, and then once we have phosphorylated the multiple tyrosines on the ITAM motifs, then the equivalent of ZAP70 for B cells is going to be called Psych, S-Y-K. So you thought it was going to be ZAP70? Psych, it's not. Oh my gosh, it's terrible. Yeah, fine. All right, and so Psych is going to be another one of these little kinases, and so there's going to be a complicated process here, of course. So the Sarc family tyrosine kinase is going to activate the ITAMs. That's going to allow Psych to bind to the ITAMs and become activated, and Psych will then be available to phosphorylate other things. Among those, Psych will phosphorylate SLP65, so different from SLP76, but a kind of a similar protein with a kind of a similar function. And then from there, PDK is going to activate AKT, just like it always does, which will then activate BTK, which will then phosphorylate and activate PLC gamma, which will lead to cutting those phosphatidylinositols into diacylglycerol and the inositol triphosphate molecule, and those will then go off and signal. So this is another way to get to the same spot, but this is how a B cell is essentially going to do it. So what do we have in common between these things? Well, we've got a whole lot of ITAMs. We've got a whole lot of tyrosine kinases to phosphorylate the ITAMs, which are the little yellow rectangles on here. That leads to activation of some kinases and some phospholipases. And the lipid kinases are going to make some inositol, multiple phosphate compounds. The phospholipases are going to both bind those and cut them to pieces. Those processes are going to lead to recruitment of and activation of some G proteins, GTPases, and some serine threonine kinases, and some phosphatases. And then down the line, finally, that's going to lead to transcription factor activation. So we've got different ways to do kind of the same thing in a different cell. And so there's really a lot in common between T cells and B cells. And you can imagine an evolutionary path where, where essentially we are duplicating the same machinery over and over again, just making a better version. B cell receptors, basically, we've figured out a way to make 2 T cell receptors attached to the same little stalk, which is neat. And the Ig alpha and beta are basically like the zeta chain, which is going to interact with the B cell receptor as the zeta chain interacts with the T cell receptor. Yeah. And then the CD3 is a later evolutionary addition, and that's going to help prop up the T cell receptor. That's the idea. All right. So, yeah, we're not going to worry about those TNF receptor super family members because, good Lord, we've learned enough. Yeah. Okay, now we need to talk about one last thing, which is co-stimulation. So co-stimulation is important the first time a T cell is activated, because we want to make sure that there is a really good signal, a really good match, and that naive T cells do not accidentally get activated, because there are a lot of T cells around all the time. It's like the same logic as if you have a gun in the house, keeping it locked in a gun safe all the time. You want to make sure the activation energy is as high as possible so that you do not have T cells activated by accident, going off and just, destroying some part of your body. Yeah. Which they are very capable of doing. And basically need to be restrained from. All right, so what are some of these molecules? Well, you got your CD28, which is going to be the T cell half. And then you got your B7 molecules, B7.1 and B7.2, which are going to be on the surface of antigen-presenting cells like dendritic cells. So this is going to be a special secret handshake that tells the T cell what kind of cell it's interacting with. Because weird as it may seem, the T cell is just running into some other cell that is expressing either class 1 or class 2 MHC with a peptide on it. And the T cell can't tell the difference except when there are co-stimulatory molecules out there. So yeah, it just, since the T cell can't see or hear what it's bumping into, this is the way that it can actually tell where the signal's coming from. And then adjust its response accordingly. All right, so on our, let's say, pretend it's a dendritic cell or one of these other antigen-presenting cells. Here's going to be our B7 proteins, B7.1 and B7.2, which are basically two copies of similar proteins like the T cell receptor alpha and beta chains. They're just going to work together and basically be two copies of the same thing. So when the T cell is interacting with MHC, and this would be true for class 1 or class 2, pardon me, oh my goodness. There we go. All right, yawn over. Then that is going to recruit a CD28 on the surface of the T cell. And the CD28 is then going to be activated. We're going to phosphorylate that thing. And a PI3 kinase will then bind to the phosphorylated CD28. And the PI3 kinase is going to take PIP2 and turn it into PIP3. And then, of course, once we have PIP3, we can do all the things that PIP3 does. We can anchor PDK, which activates AKT, which is also anchored. We can anchor ITK, which activates PLC gamma, which is bound to this complex of SLP76 LAT and GADs in the middle. Or we can activate VAV, which binds to also SLP76 GADs and LAT. But which will then activate the CDC42 and cause the cell's cytoskeleton to start to polymerize and cause the cell to start to move. Okay, so if you were summarizing the pathways, just in terms of keeping this all straight in your mind, CD28 is going to create the things that PLC gamma is going to cut. Because it's going to make the phosphatidylinositol triphosphates. The T cell receptor activation is going to lead to activation of PLC gamma. And so here's like the activation, and here is the thing that it's going to be working on. It's going to come from this pathway. And then once we've got that, PLC gamma is going to cut those little phosphatidylinositol molecules into IP3 and DAG. IP3 is going to activate a dump of calcium from the ER, which is going to eventually lead to NFAT, transcription factor binding. And the other half of that, which is DAG, diacylglycerol, is going to activate the RAS-GRP pathway, which is going to activate RAS, as you remember from that other slide. And so various kinase steps later, and we're going to end up with our FOS and June, or CFOS, C-June cofactor, which makes the AP1 transcription factor together. Each one on its own has a name, but they have a special name when they come together, like Voltron. When all the little parts come together, it's the big Voltron robot. I don't know. Yeah. Fine. And then diacylglycerol will also activate protein kinase C theta, which is going to go and activate the IKK pathway, which will then activate, let's see, it was TEC1, IKK, I kappa B, and then NF kappa B will be free to go to the nucleus and start transcription. And this is just an OCT1 transcription factor. Anyway, all these things are actually transcription factors that you can find upstream of some important genes. One of these is interleukin number 2. And interleukin 2 expression is going to be typical of T cells. So this is a way that T cells can let other T cells know that they have become activated. And so then this can bypass some of those activation steps and help to get other T cells ready to be activated a little bit more quickly and call them into the correct spot so that they can be activated. Okay. Naturally, because why not? There is a non-canonical, like a weird NF-kappa B activation pathway. And so it's going to lead to activation of the NF-kappa B inducing kinase, which is called NIC, and then NIC is just another way to phosphorylate I-kappa B kinase A, which is IKK, which then causes cleavage of NF-kappa B, and it becomes active. So this is the idea over here. We've got, you know what? Yeah, this is more than we're going to cover on the test. And so if you're interested in this, by all means look at it, but let's not go through all of that. Okay, and once again, let's jump past this. So remember your second signal. Our famous second signal is going to be B7.1 and B7.2. And that's going to interact with CD28. CTLA4 is another protein that is actually better able to bind B7 than CD28 was. So the idea is that CTLA4 and CD28 are going to be in opposition. And so they're both going to be trying to bind to B7 molecules, and binding to one will block binding to the other essentially. So if there's CTLA4 around, it will actually prevent binding to CD28. And you know that binding to CD28 is going to lead to activation, because we're going to make that phosphatidylinositol kinase activated through CD28, and so that doesn't happen. Yeah, if CTLA4 is activated. So let's jump one last little bit, and we are almost at the end now. Okay, so you've heard of ITAMs, great. There are also ITIMs, and we can go back to the thing here, which is an immunoreceptor, tyrosine-based inhibition motif. So ITAMs lead to activation. Immunoreceptor tyrosine-based activation motif, ITAM. ITIMs are the opposite. They are motifs that can be phosphorylated that will then shut down the process that has just been running. And so the idea here is that you can't just have activation. There has to be a way that we come back down after activation. And it's going to involve this thing, which is the SH2 containing inositol phosphatase, or SHIP. And SHIP is going to knock off one phosphate from PIP3. And so it's going to knock it back down to PIP2. And so PIP3, remember, is the important anchor platform for molecules like phospholipase C and AKT and all those other ones. VAV is another one. And so, yeah, if we knock it back down to PIP2, then it's ready. You can reactivate it again, but you are not going to be able to bind any of those factors at the membrane right away. Okay, so there are various proteins that have ITIMs, which look similar to an ITAM and are contain tyrosines that are phosphorylated. But the proteins that bind these are going to then, instead of being kinases, they're going to be phosphatases generally. And they are going to remove phosphates rather than adding phosphates. And so, yeah, these are some molecules that can do that, including Fc gamma receptor 2 type B1 and the KIRs. These are natural killer cell immunoglobulin-like proteins. Okay, so how can all of this work? Well, it's actually kind of neat. So how can you tell if you've actually made enough of an immune response that you can kind of back off now? If you have antibodies binding to proteins and forming a complex, such that you have multiple antibodies bound to multiple antigens all together, then that would indicate you may have enough antibodies to actually clean up the infection. And yeah, so maybe it's time to actually turn things off. So this is how that is going to work. So here's our B cell receptor, here's our Ig alpha and beta, here's one of our Sarc family tyrosine kinases. The Sarc family tyrosine kinase is going to go over here and phosphorylate the ITIM, the inhibitory motif, on the Fc gamma receptor 2 type B1. Fc gamma, remember, is binding to the Fc region, which is this part of the antibody, and the gamma means it's going to bind to an IgG, and so there we are, IgG binding to this and this, and then we've got our B cell receptor, which is derived from IgM, and so that's, but it's going to have the same specificity up here. And so if an antibody producing cell, a B cell, runs into a pile of antibodies that have been made by the same antibody producing cell, or at least are capable of binding the same antigen, then it knows that there's enough. You know, when you start running into your own comments on YouTube, yeah, you've probably been commenting enough and you should go do something else. That kind of idea. And so the next thing, so Sarc kinase is going to phosphorylate the ITIM. The ITIM is then going to serve as an binding point for SHIP. And remember, SHIP has an SH2 domain, which is why it's able to bind to phosphotyrosine. SH2 domains aren't always involved in activation. They can be involved in inactivation or whatever. They can just be binding. They can just be little anchors. And so SHIP's deal is that it's going to take a PIP3 and convert it to a PIP2. Yeah, imagine that molecule right there with a little arrow going there, and you basically got it. And now that we have no more PIP3 and lots of PIP2 lying around, we can no longer bind and activate PLC gamma. We can no longer bind and activate VAV, and we can no longer bind and activate BTK. Those guys are just shut off. Okay, wow, that was a lot. These videos are excellent. I recommend them highly. The T-cell receptor signaling is frankly a must-watch, as is the MAP kinase signaling pathway video. These are going to be things that are going to directly come up on the final, and we'll mention CD28 and co-stimulation. I think that's easier to add on once you've got the T-cell receptor signaling, but you know what? The video's there. The video's pretty good. Why not watch the video? There you go. Okay, so you made it. You made it to the end of immunology. And now all you got to do is make yourself some really good notes or flashcards or study over Zoom with a friend or whatever and get these pathways down. I would say for each one of these pathways, imagine each of the proteins is a little person with that name. And, you know, imagine some features about these people. Like Lat is long and tall and just really wants to hang out at Gad's house. And, you know, Gad's throws a great party. And so why wouldn't Slip 76 want to go to Gad's party? Slip 76, total party monster. Yeah, like that. Stupid. Again, you don't have to tell everybody else. But at the same time, if it helps you remember, it might help other people remember. So that's the challenge. That's what I'm going to leave to you. You can do this. And I believe in you. Go for it. Thanks very much. This has been the end of Chapter 7 and, the taught portion of immunology.